Thanks a lot, Christian, for the introduction. And indeed, I will try to present new results that are not published, although they are submitted. And it's a long story, so I will try not to lose you in the way and try to explain things as best as I could. And of course, well, there are not so many. If you want to interrupt me, that's fine. So you can ask questions along the way if you wish. I'm fine with that. So I will be talking about regulatory evolution, sex chromosome, and their link to speciation. So the link to speciation is really the new part of that talk because the previous work is really published already. Yes. First things first, I'd like to acknowledge my friend and collaborator, Denis Rose, who has been working with me on those topics for several years. He's working in Brittany, in Roscoff, and some funding, of course. And I'm taking the opportunity, just in case you're interested, I will be hiring some master's, postdoc, and PhD in the near future. And Montpellier is a really nice place, so if you are interested, please contact me. So when I first started my research during my master back in the 90s with Janice Briton-Davidian, who's passed away, but in her lab, she was working on mice, and I was doing cytogenetics and working on chromosome. And the nice introduction to the talk today, because the three major things she was doing in her lab, those are the three things I will be talking today. And as you can see, so the first thing is that you got sex chromosomes. So for instance, in mice, and she was looking at those chromosomes with her microscope. You got X and Y, but in many other species, you got different systems. And in mice, people also look at hybrid zones. It's very famous, in speciation, people have been looking at hybrid zone in mice. And in particular, when you look at hybrid zones, it's very frequent that you see that markers that are on sex chromosome tend to show steeper climbs than autosomal markers. And this is the so-called large X effect or large Z effect in the W species. And it's a very regular pattern that you see across the board in many species. And last, you got the famous thing that in speciation you got so-called Holden's rule. And so here is a picture showing the testes of males of mice, hybrid mice that are sterile. And it's very often the case that when you have hybrids, the heterogametic sex is suffering more, either in terms of viability or in terms of fertility, and that's called Holden's rule. And so those two major things, the large X effect and Holden's rule, are really the topic of my talk today. Although before getting there, I will try to introduce models of sex chromosome first. All right. So as you know, you got many species with different sex chromosome. Could be ZW or XY in different groups. You got completely different systems. In reptiles, it can change a lot. In mammals, they are pretty stable, and so on and so forth. It's very basic biology, you know that. And in many cases, those sex chromosomes, they show a particular pattern, which is the fact that they got recombination suppression occurring on those chromosomes, and it occurs in many cases by the accumulation of strata, meaning that different part of the chromosome stop recombining at different period, and that's something that you can document. And last thing on sex chromosome is that you often observe the fact that, especially in older systems, you got the phenomenon of dosage compensation, meaning that you've got to restore the expression levels between sexes once you got degeneration of sex chromosome. And different species have different systems to do that, to achieve that. For instance, in Drosophila, the system occurs by doubling expression of the X in male, and nothing is changed in female. That's the simplest system that you can think of. And but you got other systems. For instance, in mammals, in many cases, you got overexpression of the X, and then you got X inactivation to correct the expression in females. All right. So the hybrid incompatibilities, you know, the large X effect has been documented in hybrid zone, so thing I was mentioning before, but it's also occurring when you dissect, you look at, you know, the effect of genes when you do hybrids, in particular in Drosophila, it has been studied a lot. And this is for a typical result that you can have. I'll try to do the pointing here. Maybe, no, well, I don't know. Not working. So at the top part, you see that's the proportion of males with mobile sperm. So it's a measure of fertility. And you know, the first column represents the sex chromosome. So you do back crosses, and each time you've got the sex chromosome inherited in those cross, then you've got a huge reduction in you got sterile male. And compared to the equivalent, to an autosome of equivalent size, you see that the sex chromosome have a major effect on hybrid fertility. And that's a pattern that you find again and again. So Holden's rule is pretty general across eukaryotes, as far as we know. It has been documented in all those groups indicated on the left. And it involves either sterility or inviability of hybrids. And that's the number of cases obeying or not obeying Holden's rule. And you see it's overwhelmingly in support of Holden's rule. And you got few exceptions, few species where it's not like that. And in many cases, we know why it's not like that. It's because you have cytonuclear interactions that are making things a little bit more complex. So you got now, so that's the problem of speciation, the so-called rule of speciation that have been called like that. And now I will slightly change topic. Because people have been talking about these rules, but not really relating them to the evolution of sex chromosome per se. And here I will trying to connect the two together. And of course, you probably know that to explain the evolution of sex chromosome, you got many theories that have been proposed in the past, so 50 years or so. And the process is usually divided in several steps. So you got recombination arrest that you want to explain that part first. And here you got different theories. The most well-known is the sex antagonistic theory. I will come back on it very quickly. Then you got to explain degeneration of sex chromosome once recombination arrest is there. And here again, you got a lot of explanation that have been put forward in the past, and they all rely on some form of selective interference. And then you've got dosage compensation, where again, there are much fewer models out there about this topic, but there are a few ideas. All right. Regarding Holden's rule and the large X effect, the same. There have been a lot of theories that have been produced out there. I'm just representing the list that Cowell did recently in his paper in 2022. And you got, well, all those theories. And, well, most of them have been refuted. I mean, at least from what people usually say in reviews and what the evidence so far is pointing at. And you have three main theories today that are trying to account for the Holden's rule. The first is the dominant theory, that perhaps the most famous and the most well appreciated is the fact that it's based on resistivity of incompatibility. I will explain briefly. Then you got the faster male theory and the meiotic drive theory. So they all, they are, and the dominance and the meiotic drive can explain to some extent the large X effect, and also the faster male doesn't. So very briefly, the dominant theory is the idea that when you are in an hybrid, the difference between the heterogametic sex, or I will most often say the male and female for shortcut, the difference between the males and the females is the fact that the male is hemizygous for the X. And so if it happens to be the case that there is a gene for incompatibility, if A prime is incompatible with B, here, then the hybrid is dead. So that would be the reason it's dying. But if you are in a female, A prime is recessive to A, meaning that the incompatibility is not expressed, and that the reason the female are fine. So this idea is very, well, powerful, you could say. It does not explain well why sterility is often involved rather than variability. Why it applies to species where the female is hemizygous, in particular in mammals where you got only, I told you at the beginning, dosage compensation works by having X inactivation. So in effect, the female is also hemizygous. So Alden's rule in those cases is not super convincing as an explanation. And in fact, this theory does not really explain why those incompatibilities are recessive in the first place. It's just suppose it's like this. So the faster male theory is the idea that male traits evolve faster than female traits for different reasons, could be related to sexual selection, and so they are more easily disrupted in hybrids. Or even some arguments make it more specific to spermatogenesis, but it's really a sub-case. And so it has been very useful to explain a few cases, in particular in species where there is very little hemizygosity in the first place, for instance, in some mosquitoes. And still there is Alden's rule. So people have been saying, okay, we need something else than something based on hemizygosity. But then, of course, it does not explain very well why Alden's rule applies to ZW species and while male traits really evolve faster. So it's kind of, you can doubt that. All right. And last, you got the meiotic drive. So that's the three main competitor theory right today. The meiotic drive theory is the idea that once you evolve recombination suppression between the X and Y, it offers the opportunity for driver and counter driver to evolve very easily on the sex chromosomes. And so, and those driver and counter driver could be revealed when you do hybrids. Otherwise, that would be a reason for why you see a problem in the heterogametic sex specifically. So it's interesting because it's a process-based really model which is based on meiotic drive. But the problem are that Alden's rule often applies with viability, the first data I showed you, and viability, meiotic drive often is related to fertility, really problems during meiosis, not really viability. And of course, many cases we know how it works. It does not seem to involve meiotic drive. So today, people are kind of, well, we don't know. We got several theories, and they patch each other in different cases, depending on the situation. You say this is the right theory or this is the right theory. There is no real consensus of which prevails, and whether perhaps we need some kind of global theories where different cases patch each other. And that's what Coyne mentioned in his recent paper about this, reflecting about 50 decades of work on that topic, that while this may seem unsatisfactory to those looking for unitary explanation, we may have to be satisfied with a phenomenon that has a unitary ultimate cause, so the automorphic sex chromosome, but diverse proximate explanations. So biology is not physics, nor should we expect it to be. So it's difficult to know whether it's a regret or if it's just how it is, but there is a clear, we face this problem that there is no unitary explanation, and well, perhaps that's biology, right? So, so my talk here is that's a bit of a long introduction because there is decades of work on that topic. So I wanted everybody to be on the same page for the main ideas. And so what I'd like to do first is to present you some recent model we've been doing with Denis to revisit the way sex chromosome evolve. And so I will, it's based on those papers mentioned above. And then we will apply those models to see whether they could also explain the pattern that we see with Alden's rule and Largick's effect and so on. So the main idea is to have to explain those three sets: recombination, array degeneration, and regulatory evolution with dosage compensation. And so for the evolution of recombination, what do you need to add in a model? You first need to put into consideration that you got mutation that can restore or suppress recombination. So you could go both ways. Then for degeneration, you got to include the fact that you got deleterious mutation on genes, on coding sequences. And for dosage compensation, you've got to include the fact that regulators have to evolve. And you got to include that. That's a minimal ingredient if you want to address the evolution of sex chromosome. And so the idea was just to bring together those key ingredients alone in a single model. And comparing to previous work, in particular, the last part, the fact that we include regulators was not done before, and it actually changed quite a bit the outcome, as you will see. So that's mostly something we have been working out in simulations, including many loci. And so the way the model is built is that we consider chromosomes where you got many genes, or say 500, and they are on a map, so you got linkage. They are equally spaced along the chromosome, and on the chromosome you also have a sex-determining locus with the X and Y allele determining whether you are in a male or female. And we work in a relatively large population sizes. Of course, you have been exploring different values, but it's a large population sizes. Then, of course, you've got mutation, deleterious mutation occurring on the genes. So you got with the standard population genetics effect, like when you got when you are homozygous, you are 1 minus S, heterozygous 1 minus HS, where H is the dominance of the mutation, the deleterious mutation, and on average, the H is 0.2, 0.3, depending on estimates. They are partially recessive, and that's what we use. And the S, we consider an exponential distribution such that we have a lot of small effect mutation and few larger effects. And importantly in that model, because the mutation need to add up to have degeneration, then we consider that the effect can pile up on a gene up to, or the additive in an additive way, up to a value which we set here to be 0.3, meaning that the gene is not functioning anymore. It has a 0.3 effect on fitness, which is a huge effect, right? But you can change that number. It's not dramatically changing anything. Okay. Then for recombination is a bit more tricky. We've been trying different versions of that model, but the simplest way to think about it and to handle the situation is to consider the fact that you first draw something that you could think of as being inversions. So you sample in a uniform distribution the size of the chromosome that would stop recombining as if it was an inversion, and it occurs in a given individual on a given Y, and it occurs at a given rate, U, E, and V. And then if you got a second one, then it adds up so that chromosome recombination suppression can extend along the chromosome. But those things can be reversed. I mean, it's not a one-way street. You can have a reversion event such that the last event can be restoring recombination. Can discuss other. We've been trying other systems, but this is the simplest one you can think of. And then we measure through time the length of the chromosome which is not recombining. And the last part, which is perhaps the most novel with respect to past theory, is the fact that we consider the evolution of regulators. And to do that, we have in mind the fact that you want to let the model evolve dosage compensation, because that's really the key to the evolution of sex chromosomes. And to do this, well, we just mimic what's going on in eukaryotes, the fact that you've got both cis and trans regulators. And so cis regulators can be, so that the orange box, so you can think of them as being like promoters or enhancers of a given gene. It means that if you have a mutation in that cis regulators, it will change the expression level of the gene the coding sequence on the same chromosome only, not both. Whereas, and then you've got transacting factor, which are, you can think of as being transcription factors, and they are expressed. They, for instance, can be protein, and those protein, they are, diffusible molecules in the nucleus so that they can interact with both cis regulators. So if you got a mutation in a transacting factor, it will impact both genes, homologous gene. And that's a key difference. Cis regulators are asymmetrical because they only influence the copy next to them. Transregulator are not, right? They can influence the overall expression level, but not the difference between the two copies. And of course, we need that you got transcription factors that are expressed in males and females separately for each gene, such that dosage compression is completely free to evolve in males or female completely freely without any asymmetry in the model or without any constraint. Okay. And okay, so then you've got mutation on those regulators, and they are quantitative traits. So they got values as a trait. They've got normal deviate for mutation effects. And then you add up those effects, and they are additive. An important thing is that when you've got the two trait value for the cis regulators, imagine you've got one cis regulator which has a stronger trait than the other one, then it creates an imbalance between the expression levels of the two genes. The one with the strongest cis regulator will be more expressed relative to the other. And as a consequence, we have to tune the fact that it will change the dominance level of a deleterious mutation that will be on that gene. For instance, if something is not expressed at all, then the deleterious mutation that would occur on a gene there would be recessive. And the transregulator, as I was telling you, is the same, is also a quantitative trait, additive quantitative trait. And it just changes the overall expression level that you can get at a given gene. And those expression levels are all under stabilizing selection, such that there is an optimal value for each gene, the same in the simulation that you target, the same in male and female, and you just let the system go. All right, that's so a typical output of a simulation like that when you put that kind of model. So I can walk you through this figure just so that's on the x-axis you've got generation, so number of generations. So that's a long simulation, 10 million of generations, so it takes some time. On the y-axis, you've got the z value, that means the fraction of the Y chromosome which stops recombining. And it occurs because you've got the fixation of some inversion of different sizes, which were randomly drawn, as I explained before. And each time you've got a fixation of such an inversion, pretty quickly after that, you got the evolution of the gene on the Y that becomes silent. So that's the green curve. That's the fraction of the genes that are silent on the Y. And in red, that's the fraction of the genes that are knocked out, meaning that they accumulate deleterious mutation to 0.3, the maximal value we decided it was knockout. So meaning that the gene, and you can see that all the genes in each stratum actually degenerate completely, and that this part of the chromosome becomes silent pretty quickly. Well, pretty quickly, it's a matter of, you see the time frame. And then you got this accumulation of different strata, and the system is pretty stable. It does not come back, right? Once you evolve a stratum, it stays there. It does not come back. Even though you've got a very high rate of reversion in that kind of model. And so you can dissect what's going on. And in fact, you got in green, you got some of those inversions. The reason why they I won't be able to give all the details because that's not the point of my seminar today. But the reason why they get fixed in the first place is because they tend to be lucky. They catch a portion of the chromosome which is by chance with fewer or less harmful deleterious mutation. You've got the segregation segregating variants in the population. And so you can sample a chromosome which by chance happened to be lucky in that sense. And that gives them a selective advantage initially. That's why they raise in frequency. But then they also directly start accumulating the latest mutation. So the question is why they are not reversed. Some are reversed in red. They fix, but then they become reversed. But some don't, and some stay around and are never reversed. So why is that? Oh, before that, and then you can see that the dominance level from the point of view of gene on the Y. So the dominance level dropped to 0 because it becomes silenced, not expressed. And that's the accumulation of the latest mutation of on average for each gene in each stratum on the bottom graph. So they accumulate a lot of mutation. So why is it that the reversion in that model are not selected for? That's the key thing to understand, because that the key insight of those models, in fact, is the fact that so this figure show on the x-axis the inversion age since it first occurred, and in the y-axis it's a marginal fitness of that inversion compared to the same fragment of the X chromosome in the population. And so initially, as I was explaining, you see that those inversions are lucky to begin with, meaning that they got a marginal fitness which is higher than average. So that's the reason they tend to selectively fix in the population. But then it starts, their marginal fitness tends to decay because they accumulate the latest mutation pretty quickly. But before they reach fixation, but then they reach fixation. And once they reach fixation, you know, the cis regulator on those inversions start to diverge, such that you know, you got this silencing of the Y that start to emerge. And once you get there, in fact, the marginal fitness of that inversion becomes higher than the corresponding fragment of the X chromosome. And so the reason for that is the emergence of dosage compensation, as I will try to explain. So the two things that occur in that model are different from the classical theory. First, degeneration occurs, but it's not directly linked to selective interference. It's not due to the fact that selective is less efficient due to the lack of recombination. It's due to the instability and the divergence of the cis regulators. You are in an unstable equilibrium where, you know, you got diploid expression that is controlled by the cis regulator on the X and the Y, and it can go either way because it can start to diverge. And it's an unstable equilibrium because you can go, but you can only go in the direction of silencing the Y. The reason is that if you start silencing the X can increase in frequency, but then when it becomes homozygous, it will reveal the deleterious mutation. So it cannot in female. So it cannot go in that direction. So the unstable equilibrium is always pushing the towards Y silencing. So it's a different explanation. It works for one locus at a time. You know, it does not rely on selective interference. So it's a different model for degeneration. And so the reason why the system is stable is the fact that when you start evolving dosage compensation, it creates sex antagonistic effect automatically, if you wish. Meaning that imagine you bring imagine, so you got this Y, which is with this inversion that start to be silenced and accumulate deleterious mutation. You would say it's pretty harmful for the fitness of male to have all those deleterious mutations, and maybe it would be worse recombining to bring from the X fresh, clean version of the genes, you know, to restore male fitness. And that's true, except that when you do so, when you bring you got a reversion event, you bring by recombination some gene from the X back onto the Y, what's going on is the fact that you bring genes that are supposed to be fully expressed in males that are started to evolve dosage compensation, meaning that they will overshoot their expression. And the reverse occurs in the other direction. For each gene, the emergence of dosage compensation creates a sex antagonistic effect that builds up and that prevent recombination to be restored. Okay, so that was very quickly the stories that we've been publishing, trying to explain that models of sex chromosome could be seen differently once you put the evolution of regulators into the picture. And so what the remaining time I'd like to explain is what it does to hybrids, which is a new part, if you like. So you take the same model, and now you got independent replicate, and you consider that it's different as if it was different species, and you make hybrids, and you look at the fitness of those hybrids. And so that's a typical outcome that you see. So on the x-axis of that, so on the x-axis of the figure, you will have the fitness of female, let's say, and so the homogametic sex, and on the y-axis the fitness of males, and the dots are from pairs of hybrids that you create between different pairs evolved independently, and you do so at different time point. In orange, that's pretty early on. That's that's 250,000 generation. In red, that's after 1 million generation. And in a darker brownish color, it's after 4 million generations. So the line here represent the fact that male and female hybrids have the same decrease in fitness. Okay, that's not Alden's rule. Alden's rule is when you expect the male fitness to be what? The fitness of the heterogametic sex to be lower. So that's what you see. So in color, so that's what you observe in those model, and in the gray dots is when in those simulations you start with a Y chromosome, which is just with the sex locus. There is no recombination arrest. It's just it's a homomorphic. There is no strata, nothing. And in gray is the model when there is no inversion, there is no recombination suppression that can evolve. So in fact, what you see is, and if you run simulation with autosome, it would be the same. So you do observe a decrease in fitness due even on autosome in both sexes, but it's pretty small. And it's due to the fact that the regulators, cis and trans regulators, diverge a little bit between replicates. And so the hybrids are less well adapted. But the key thing is that when inversion can evolve, when recombination arrest can evolve, you see that you've got a major decrease in fitness in males or the heterogametic sex. And you can do the same. So that's the same kind of plot, except that instead of dots, I've put some regions of the where you got the dots to have a lighter figure, and you can start with different starting points, whether it was a completely new Y, whether it's a Y which is already half degenerated in the ancestral of the two species, or if it's a XO system, meaning that there is even no Y, right? Some species show Alden's rule and there is no between XO species. And in all cases, you see this phenomenon where you got a dramatic decrease in fitness for the heterogametic sex. So why? Well, in fact, there are two reasons for this pattern. And the first is, well, they are related but slightly different. For the first one is for the genes that are in an ancestral non-recombining stratum that is shared between the two species. So in that case, meaning that in that situation, the two, if you like, the two Y's they start being already degenerated and dosage compensated, but in the independent replicate, the cis and trans regulators will start diverging. And what's going on when you do the hybrids is that in female you will have, so it's so the regulator trait are additive, but so you will have average the trans acting traits in female, and you will also average the cis acting traits. Both the cis and trans traits are averaged in females, but in males you only average the trans trait, not the cis, which is only expressed from one Y coming depending on the direction of the cross from one species or the other. And so this problem of averaging decreases male fitness more than it does in female. And for the genes that are in a derived stratum, meaning that they are only present in one of the two species, while the situation is perhaps intuitively clear, it means that in one species the genes are just working well, and in the other species, one is degenerated and dosage compensated. So depending on the direction of the cross, you will have in the hybrid a degenerate gene with half the dosage compensation, or you will have a fully functional gene, but also with half the compensation, so it will lead to overexpression. So now perhaps a little point about viability versus fertility, because some patterns that have been repeatedly mentioned in the problem of to explain about Holden's rule. So here, what you can do is it's difficult to represent in that type of model what would be a viability versus a fertility gene. But what you can say is that for fertility genes, there is a chance among those genes that some are sex specific in their expression. Perhaps they are expressed in the testes, on the ovaries, in the germline of the different sexes, and they are different genes. So what you can do is to consider those genes with a sex-limited effect. And if you do that, then you find a very strong pattern of Holden's rule as well. But here, so you got two cases that are a bit different. In the first case, what is called the sorted case, it means that you decide. First, you got to realize that when you have sex-limited genes, in female, it does not change things very much because in any case, you got a gene for fertility on the X, it cannot degenerate because you need that in female, and the female is XX. But in males, if you got a fertility gene which is only expressed in males, it can also it will degenerate, but it can degenerate either on the X or on the Y, depending on. Well, it can go either way. Contrary to a gene that is expressed in both sexes, fertility genes like that can be lost actually on the X, and not only on the Y, as you know the other cases were considered before. And so, depending on the replicates, you can lose the gene either on the X or on the Y. And so that's the unsorted case at the bottom. So it creates massive deleterious effect for fitness, because in the hybrids, you will, in some direction of the cross, you will completely miss the gene. And so the male will be completely sterile. But even if it's already sorted out, meaning that the two species share the two genes that are already on the ancestral species already divergent, and it means sorted, that is, it's already decided which copy degenerate, whether it's on the X or the Y. The effect also is larger than before, because here the gene degenerate on the X, only evolve on the Y, where with a very low effective population size. So I know it's a lot of information, and because this model are a bit difficult to really explain in all details. Of course, I will address if you've got questions. But what I'd like to point out to finish is the fact that so those models link directly the evolution of sex chromosome with the observation of the rule of speciation, meaning the same model predict both phenomena without added ingredient. So you got a large decrease in fitness of the retrogametic sex in F1 hybrids. You got a large effect of the X, because that's the only chromosome compared to autosomes that evolve dosage compensation. You got a larger effect than average of fertility genes, for the reason I briefly mentioned. It works in species with any kind of sex chromosome, whether it's an old sex chromosome pair or a very recent one. And it's pretty quick because, as I was showing, it's, well, the order of millions of generations only. Then there are many studies out there starting to document the fact that there is a lot of misregulation of gene expression in F1 hybrids, that many speciation genes are related to dosage compensation. And there are issues that I don't have time to get into, but so maybe we've got a single model here. At least we can consider the prospect to have a single model that would explain recombination arrest on the Y, degeneration, evolution of dosage compensation, evolution of this one I didn't talk about, evolution of somatic versus germline dosage compensation, Holden's rule, which was the main topic here. And so Darwin's corollary to Holden's rule is the idea that depending on the direction of the cross, when you study Holden's rule, one direction of the cross is more severe than the other, and that's something you get all the time in those models because it depends on whether the Y is degenerated or not in the two species. Of course, you got a large X effect, a disproportionate effect of sterility gene in Holden's rule, and last, you got this idea that briefly mentioned that on autosome, you also have a signal of hybrid breakdown, so at a slower pace, but you also expect that after time goes on, you accumulate incompatibilities for all genes, even if they are autosomal, because of the coevolution of cis and trans regulators, so that eventually, at a slower pace, you will eventually lead to a situation of complete reproductive isolation for all genes. In fact, if you wait long enough. So to conclude, so that was to answer perhaps Coin. Maybe there are simple laws in biology, and perhaps the take-home message I'd like to see is that the problem is that there have been so many researchers trying to work on those things, many theories proved and disproved, and maybe this one will be disproved. But and I think in biology the difficulty is that we need to look at so many planets. You know, it's not like in physics, you just have 8 planets in the solar system, but so many to look at before we can even try to look at patterns and to see what is it that we need to explain in the first place. And that's something which makes it even more challenging to compared to physics. And with that, I will be happy to take questions.